Okay, thank you. That's a hard act to follow. I want to thank the organizers for putting this together. I'm very excited for these next few days. I'm going to talk a little bit about some of our work to animate coacervate materials with metabolic or enzymatic activity. And before I dive into what I'm going to talk about, I want to acknowledge the people that actually did the work. Okay, well, I can gesticulate wildly. The work that I'm going to share today was largely done by a single graduate student, former graduate student, Nisha Modi, as well as some help from current students, Jane Liao and Femke Janssen. And all of this was done in collaboration with one of my wonderful colleagues, Kyle Bishop. My lab is interested in protein complex coacervation, so I'm a chemical engineer and really a chemist by training. And so we like to think about how proteins undergo this liquid-liquid phase separation with oppositely charged polyelectrolytes, this process known as complex coacervation, and what changes when instead of two linear Gaussian coils, one of your polyelectrolytes is a folded globular protein. And so we've studied this quite a bit in vitro, and then in increasingly complex mixtures that maybe approach some of the things that are of interest to those in the audience. So we've looked at how the phase behavior changes when you don't just have a binary mixture of a protein with an oppositely charged polymer, but in fact a handful of well-defined either proteins or polymers. And then getting into more cell-like systems, we've looked at protein complex coacervation with endogenous biomolecules and cell-free lysates and inside of living cells. So as an example, today I'm going to talk about our work in vitro in a talk that I have never given before that I'm hoping is of interest to this audience because it does bridge experiment and theory. And so as an example, here is a supercharged GFP with a net charge of -12 that's distributed randomly over the protein surface. This forms these liquid fluid-like droplets with a linear polylysine polypeptide with a net charge of 30. And we can look at how changes in the protein sequence impact the material properties or rheological properties of the complex coacervate materials using, here I'm showing you a video particle tracking microreology. And over the timescales that we can monitor particles with a microscope, we see that these complex coacervates are viscous fluids. So you see here the mean square displacement, and then the storage and loss moduli. But what a almost former postdoc in the group did was adapt a particle tracking technique that uses dynamic light scattering, which allows us to report on the rheological behavior over a much broader range of timescales. And what we see now, when we look at the complex moduli plot, is that what we saw by video particle tracking microreology was a viscous fluid, but in fact, this is a viscoelastic material. You can fit with a simple two-component Maxwell model. And this looks, if you study complex coacervates of linear polymers, this looks almost exactly like the rheological behavior that's seen in those materials. Okay, so this is the equilibrium. So we're interested in the equilibrium behavior of these complex coacervate materials. But what I'm going to talk about today is these materials out of equilibrium. And I will say, I'm very nervous to do so, because a lot of this was inspired by some of the incredible work of people in this room. So I don't know how many of you were inspired by these beautiful papers from David, and showing that these active droplets resist Ostwald ripening, and at the right conditions, they can grow and instabilities can form, resulting in droplet division. And so we saw this paper right when my lab got started. Our minds were blown, and we're like, can we do this in vitro? And so we're obviously not the only ones that are interested in active coacervate type materials. So here's work from Eric Dufresne's group, now at Cornell, where they take a simple coacervate, in this case of BSA, and a high concentration of PEG. The enzyme urease goes inside, and the enzyme activity The urease generates flows, which causes the droplets to swim toward each other via a Marangoni type effect. Okay, there are other ways for droplets to be out of equilibrium, or these Coacervate droplets to be out of equilibrium. So here, work from Tom Knoll's group, in collaboration with David Waits, I believe, showed that simple Coacervates, if you rapidly change their temperature, can form these internal vacuoles. They showed this both experimentally, as I'm showing these microscopy images on the top, and as well as in simulations. And they more recently showed this also in complex Coacervate materials, not necessarily, they do also show vacuoles, but that's not what I'm showing you here. So they have these multi-layered complex Coacervates. We have a dense core and a less dense second phase on top of that. And again, if they rapidly change the temperature, they can generate droplets of the other phase inside of the other phase, and leading to a lot of new interfaces. Okay, and this increase in the interfacial area can result in faster cargo uptake in these complex structured droplets. Okay, so just to say, we're clearly not the only ones thinking about this, the only ones interested in this. There's obviously a ton of other beautiful work that I don't have time to talk about today. Before I start sharing some of our work in this space. And so we are thinking about using these simple complex Coacervates as a model for active cellular condensates. And so what we do is take a weak polyion. So in this case, the charge is going to be pH dependent, which is what a weak polyelectrolyte is. And then we form a complex Coacervate with an oppositely charged enzyme, where that enzyme's enzymatic activity can cause a pH change. Okay? And so in this way, we hope that we can couple the enzymatic reaction to a pH change. This happens naturally. And then this pH change will be coupled to phase separation, which can then potentially impact the enzymatic activity. Okay, so that's what I'm going to tell you about today. I'm going to tell you two different stories where we're looking at different rates of pH change. Okay, so if we have very slow rates of pH change, we're effectively maintaining equilibrium. If we increase the rate of pH change, where this rate relative to the size of the droplet is faster than the diffusion of the polymer, we think we can get vacuole formation. And then when we have very fast pH changes, where the rate of pH change is fast relative to the diffusion of ions, we think maybe we can achieve size control. Okay, so first I'll talk about these fast rates of pH change. And for this system, we used the weak polyelectrolyte DEAE dextran with the enzyme catalase. And these can form a complex Coacervate at neutral to basic conditions where they're both charged. Catalase can cause a pH change due to the decomposition of hydrogen peroxide. So this decomposition of peroxide then is coupled to the acid-base equilibrium of the peroxide molecule itself, which then degradation of peroxide results in a pH increase in the solution. Okay, so then we can potentially get reaction-induced droplet dissolution by adding the enzymatic fuel or the substrate to solutions containing the droplets. Okay, and then again, if our droplet size is greater than some critical radius, A star, then we can get gradients in the pH inside the droplet versus outside the droplet, driven by the enzymatic activity, which potentially could lead to size control, where droplets greater than this radius now are unstable and should disassemble. Okay, so the first thing we did was characterize the pH change that we could induce with this enzyme. So all of this, what I'm showing you here, was done in a very small concentration of buffer to keep the enzyme functional and soluble for the day that it required this student to do these experiments. And we can see that we can get a pH change from effectively nothing at basic acidic pHs to about 1.5 units at basic pHs. We can fit this with a simple model for the enzymatic reactivity, and it matches as long as we include the fact that there's a very tiny amount of buffer in there, like less than 1 millimolar, I believe. So if you're an experimentalist. And so from this data, we chose to operate at high pH such that we can drive a large pH change with the addition of substrate. We also looked at how substrate concentration at this pH impacts the pH change. And again, we see a concentration-dependent pH change, again, with optimal pH change effectively saturating around 100 millimolar of the substrate. And so we chose this lowest concentration of substrate such that we can hopefully minimize the formation of oxygen bubbles, which is oxygen is a product of the hydrogen peroxide decomposition. Okay, and then just to show you how quick this process is, here is a pH measurements of the unbuffered solution with the enzyme catalyzing this decomposition. When we don't add substrate, we see effectively no pH change. And with the presence of hydrogen peroxide, within, I don't know, 5 to 10 seconds, we now have a 1.5 pH unit pH change. Okay, and so we define a characteristic time scale based on this rate of change. Okay, so now we have an enzyme that can cause a pH change. Do we have a system where we can have droplets or phase separation depend on the solution pH? So what I'm showing you here is we're just varying the ratio of enzyme and polymer to identify the optimal condition to mix them to drive complex coacervation. This should be optimal at charge neutrality, which is where we approximately see a peak in this turbidity indicating in vitro phase separation. Okay, then we can take at this specific mixing ratio and now look at different pHs and look at the turbidity of the solution, or look at the droplets by optical microscopy. And we see that we observe phase separation between, let's say, pH 7 and 10. And as we go to higher pH, these droplets start to dissolve. So we don't see droplets at pH 11. And so we're hoping that this 1.5 pH unit change caused by the enzyme can drive droplet dissolution. Okay, so now in theory, we have two of the components to couple enzymatic activity and phase separation. So here were our initial attempts to look at this. So what you can see on the top is a synylation vial containing this enzyme with the polymer at pH 9. In the absence of buffer, you can hopefully see that this is turbid. And then down here is a graph of this data from multiple replicated experiments. And you can see that it has high turbidity, and then the pH is around pH 9. Okay, once we add the hydrogen peroxide, you can see that the turbidity drops somewhat significantly, and the pH increases to about 10.2. In the presence of buffer, we still see a turbid solution at pH 9. But even when we add the substrate, because we're now controlling the pH with buffer, the pH doesn't change, and the turbidity of the solution doesn't change. Okay, so then what we've shown is that we can potentially have this two-way feedback where the enzymatic reaction affects coacervate formation. And then we're interested to know if the coacervate droplets affect the enzymatic reaction. So to look at this, we monitor the pH change over time for a variety of different situations. And so what you see here in these first two, so you see one trace here and then multiple experimental replicates with this characteristic time scale in the graph here. And so the free enzyme in solution is virtually indistinguishable from coacervate droplets, indicating that just being in the coacervate itself does not seem to impact the enzyme activity, at least in a significant way. But then if we collect all of the droplets by sedimentation into a large droplet, okay, it's like a millimeter, then this activity is, the rate of pH change is significantly slower, but it's indistinguishable in between this pellet and supernatant and then just the supernatant, which is indicating that all of the enzymatic activity is coming, that we observe in this case, is coming from the supernatant, indicating that we have massive diffusional, well, our interpretation is that we have massive diffusion limitations in the large droplet, such that effectively we have all reserved enzyme activity in the droplet, which is not actually contributing to the pH change. And so we, largely my collaborator, built a simple reaction diffusion model that relates the apparent rate constant to the drop radius using very standard chemical engineering methodologies. And so here we can see that we basically reach a critical radius above which we now start to see that the activity is limited by the fraction of enzyme that's in the dilute phase and this rate constant. Okay, and then again, this critical radius is related to where reaction and diffusion rates are balanced. Okay, so then with this model, if we can have constant external conditions for small droplets below this critical radius, we see homogeneous distribution of substrate throughout the droplets, homogeneous pH throughout the system. However, when our droplets are significantly larger than this critical radius, now you can see that the substrate does not reach the core of the droplet, again, as you would expect from a basic reaction diffusion model, and then that generates high pH on the inside of the droplet, which should lead to an unstable droplet. However, if you were eagle-eyed, and when I showed you those scintillation vials, did anybody see anything weird? Yeah, like half the container was filled with bubbles. Okay, and so that prevented us from, despite all of our very hard efforts and all our niches, very hard efforts, we could not observe the droplets undergoing the reaction. The amount of oxygen, we tried everything to scavenge the oxygen. The amount of oxygen being produced was just simply too much to effectively remove. And so we couldn't try to see if we could set up this non-equilibrium steady state. The other thing that we did build a chemostat to control the pH and glucose inlet feed. But again, even in that chemostat, the bubbles were too, just like violently bubbling. We just could not track that system by any method because there's so much interference from the bubbles. So you do, in order to get this non-equilibrium steady state, in agreement with what others have shown, is that you need different reaction rates inside and outside of the droplets. Here we can achieve that just by having increased partitioning of the enzyme in the complex coacervate. Of course, to achieve this instability, you should have fast reaction rates inside of the droplets relative to the diffusive exchange. And then you do need, a non-steady state condition is not going to lead to this droplet size control. Okay, so in this system, at this point we said, okay, let's pivot to another system where we don't have the problem of gas bubbles. And so this, as I'm going to tell you about now, this is currently a preprint. And so to do this, we simply changed from the enzyme catalase, which is very fast, to the enzyme glucose oxidase, which is pretty slow. And so this still phase separates with DEAE dextran. In this case, we're starting at an initially higher pH because the hydrolysis of the product of glucose oxidase is much faster at higher pH. Okay, so here, hopefully you can see these droplets. Hopefully the contrast is sufficient. And then we can drive a pH change and potentially see uniform droplets or droplets with vacuoles. So if this pH change is slow, you see these droplets look as they did initially. The color is different, and I will explain that next. But if this pH change is fast, now you see that we have these internal vacuoles inside of the droplet. Okay, if you're not convinced by this image, we also fluorescently labeled the enzyme and a confocal microscopy, and you can see what initially is a relatively homogeneous droplet then has this large vacuole after a fast pH change. Okay, and so we saw these differences in between fast and slow pH changes and what happened to the droplets. but we wanted to be able to quantitatively track this. And so we set up these capillary experiments where we can load a high concentration of acid or substrate at one end, and then watch it diffuse over the length of the capillary. In this case, you can see the diffusive front because we've simply added a pH indicator dye. And so the indicator is xylenol blue, so it's blue at pH 10 and yellow at pH 8, maybe 7.5, and red actually at pH 3. So that's two relevant pKs, and so you can see over time, you see this acid diffuse through the capillary. Okay, and if we load the capillary from the other end with a solution containing Coacervate droplets, we can then monitor them over time using a color camera, and we can infer the surrounding pH. So we can convert this color image into the XY chromaticity, and then use that to estimate the surrounding pH. And so we can see here, looking at an individual droplet, initially near pH 10, as the pH decreases, you start to see these inhomogeneities inside the core of the droplet. As the pH continues to decrease, you see these grow. You also see the droplet swells. Okay, that's for one droplet. We can also watch this in real time, where on the left you see the image of the droplet. On the right you see our instantaneous determination of the pH, as well as tracking of the droplet radius. Okay, and you'll see, hopefully, it forms these vacuoles and swells and grows. Okay, so with these movies of droplets, now we can quantitatively track their behavior over time. So for each droplet, we can infer the pH based on the color of the pixels surrounding the droplet. And you can see here that as we diffuse in acid, the pH decreases monotonically. We just define a reference pH at pH 8.5 and estimate the rate of pH change more or less by fitting a line to this pH. Not exactly, but I'm happy to talk about that. And then we can identify when vacuoles form from the video. Okay, so we can do this for each droplet, but we can also image at multiple lengths along the capillary to look at different rates of pH change. So basically, the further away from the acid source we are, the slower the rate of pH change will be. So we just image at multiple locations and then track the pH, rate of pH change for all the droplets. So that's what I'm showing you here. We've analyzed several hundred droplets this way, and we can have rates of pH change that span slightly more than an order of magnitude. You see 4 different color lines. That indicates 4 different experiments, and the gray is the summation of them all. Okay, we can, right, I said that. So we can also then quantify the droplet size. And so we see a non-monotonic change with pH, where initially the droplets seem to shrink before rapidly swelling. And we analyzed all droplets that were bigger than a critical threshold because we could not, we did all this analysis in an automated fashion, but we also checked every single droplet in the call that our automatic detection made, and we could not visually confirm droplets below 3.4 microns what was happening. And then we used the angular asymmetry of the droplet to identify the point of vacuole formation. And so basically the droplets are homogeneous, and then once they have some irregularity, that should result in an increase in the angular asymmetry. And so again, looking at tens of different droplets, we just defined a threshold of 1.2, and above that, we called those as having formed vacuoles. And then from this, we can see that the pH at which vacuoles form varies across these different experiments over about 2 pH units. Okay, as you might expect, if you are familiar with reaction diffusion models, this depends on the rate of pH change. Okay, first it depends on the size of the droplet. So large droplets will form vacuoles earlier, okay, because diffusion should be slower because they're larger, and so these form vacuoles earlier. And then for droplets of the same size, a faster rate of pH change also results in vacuole formation earlier. Okay, so I've shown you now 4 droplets where I made that claim. But again, we can analyze the hundreds of droplets from these different experiments. And we see that the trends are consistent over multiple droplets and multiple experiments all done on different days over the course of, I don't know, 6 months. Where you generally see the trend that as you either have a larger radius or a larger rate of pH change, a faster rate of pH change, you form droplets at a higher, form vacuoles at a higher pH. Okay, so we can look at just one of these 2 factors and see over a narrow range of rates of pH change, we see a strong dependence of the pH at which we form vacuoles on the size of the droplets over an albeit relatively modest range of drop sizes. And we can do the converse where we look at a relatively narrow size range and all the different possible rates of pH change. And again, we see a strong correlation with the pH of vacuole formation on the rate of pH change. Okay, so this is all experiment, and this is supposed to be bridging experiment and theory. So this is going to be my attempt to do that very quickly in 2 minutes. And so we developed a minimal model that describes the pH-dependent phase equilibrium. This is a simple 3-component Flory-Huggins model where we have polymer, enzyme, and water. And then we simply have a pH-dependent chi parameter. And so we have an electrostatic component and a non-electrostatic component. And we chose parameters to approximate the densities or the concentrations of the enzyme component in each phase at pH 10. And then these tie lines should approximate charge neutrality, with the slope being the ratio of the charge densities of the 2 polyelectrolytes. Okay, and so for a slow pH change, this is a quasi-static path, where effectively you're just moving along the binodal as you go from one pH to the other. But then rapid pH changes resulted in spinodal decomposition. And so to look at this, we used a Kahn-Hilliard approach, again, the simplest model that we could use to derive the time evolution of the mixture. And so what you see here is a fast and slow mode. So, okay, we're using, again, all the standard equations for the diffusive dynamics. We see that there are 2 relaxation modes. So the first one is establishing charge neutrality. And the second one is effectively exchange of charge neutral complexes due to the density difference at the 2 different pHs. Okay, all right, I have 1 minute. So this is a simulation of this happening. So again, you see somewhat similar behavior to what we saw in experiments, where we see this initial expulsion of material and then coarsening of the vacuoles within the droplets. And then with this model, we can run simulations at various sizes and rates of pH change. And again, in agreement with experiment, we see that there's a region of vacuole formation with relatively fast rates of pH change and relatively large droplets. And there seems to be a critical droplet size below which we don't form vacuoles. Okay, and so this black line corresponds to the slowest diffusive relaxation rate, and then the horizontal dashed line indicates the spinodal growth rate. Okay, and so there's 2 requirements for vacuole formation. The rate of pH change needs to be faster than diffusive equilibration, so that you have this non-equilibrium condition. But you also need the rate of spinodal instability to be faster than diffusive equilibration to form these vacuoles. We then did some FRAP experiments, which allowed us to then scale our experimental data and match this with our simple model, where again, this is the same black line of our slowest diffusive mode, where we've now reduced the rate and radius by experimental parameters. Okay, and so now we're going towards reaction-driven pH changes. Here's a video. a little slow, and I know I'm, okay, 1 minute. You could give me 1 minute, please. You see these vacuoles grow, and this is just with the addition of glucose. Unfortunately, in the absence of indicator, so we don't know the pH, but you can see these vacuoles form, not when just acid is diffusing in, but when we have enzymatic-driven pH change. Okay, so what I've shown you, hopefully, is that enzymatic activity can be coupled to complex coacervate formation, and then internal or external destabilization drives vacuole formation, and this vacuole formation depends on the drop size and the rate of pH change, and with relatively simple models, we can qualitatively describe vacuole formation. What I'm not showing you is we can do this with other polymers and other proteins and observe similar behavior. Okay, so with that, I'll thank everybody again and have time for questions.
Thank you for the great talk. I'm interested in the morphology of these vacuoles because it seemed as if they were touching in your experiments, whereas in the theory they were like nicely separated droplets and coarsened over time. So do you know anything about the material properties?
Yes. So this is something that we, great question. We have tried to measure the rheological properties at the different pHs. The droplets we can form at pH 8 are very small, and they nucleate very slowly. And so we haven't been able to look at that. But we do think that the material properties may also be changing over time as we change the composition. And we do not capture that at all in our model.
Back.
Yeah, a question about the vacuole formation videos, especially the experiments, look very interesting. But the theory models one droplet, whereas in the experiments you have other droplets around. Do you think that might contribute to some of the different effects you see? Certainly the other droplets should act as sinks, I guess, of your substrate as well.
Yes, that's a good question. So that's a good question. So I think in the case of the catalase, where the process is so fast, that may, based on the distance in between our droplets, that may be relevant. But I do think the size of our droplets, the distance between them, and the rates of pH change that we're seeing, I don't know how much they're coupled to each other. But yeah, it's something we haven't tried to explicitly, we did different experiments where we explicitly tried to get, faster rates of pH change, slower rates of pH change, smaller droplets, bigger droplets, but we haven't tried to do anything like, patterning the droplets at different distances from each other, but that could be interesting.
Thanks for the nice talk. A quick question. I was interested in the experimental protocol. You showed a response to a change. Have you ever thought about introducing a time scale of glucose wash-ins and wash-out, or fuel wash-in and wash-out, and whether you get new non-equilibrium morphologies when you oscillate your external field as opposed to having a pulse?
Yeah, good question. We have thought about that. I mean, so I would say we've largely thought about this single wave versus a steady state chemostat, but in the chemostat we could certainly oscillate the conditions, and yeah, that would be interesting.
Hi, quick question. The droplets inside the droplets and the large droplets themselves, they're growing by ripening or by coalescence, or is it a combination between the two?
Yeah, good question. So we haven't looked at the time evolution of the vacuoles and how those change in size. So I can't say. We do think that they are coalescing, but that's just based on looking at the videos and not actually fitting anything to model their.
Okay, wait. Sorry, good to you. Quick clarification. Then we have Hangbo, Frank, and then Ben.
Sorry.
Another quick follow-up question. And the size control you observe is happening on the bigger droplets or the smaller ones?
So we don't yet see size control because that was we need faster rates of pH change to generate gradients within the droplet, we think. And so glucose oxidase, the rate of glucose oxidation is actually not that slow. It's the rate of hydrolysis of the lactone to the acid that is slow and pH dependent and slows down as we change the pH, as it's changing the pH. So that process slows down such that we need the critical radius for the glucose oxidase droplets to try and set up this size control is on the many tens of microns, and those are really large droplets. We haven't yet looked at them or tried to make them that big.
When there's a pH gradient, like the HCl, you see any swimming behavior because you cited the Eric difference, and also toward the end, you were showing a movie where the droplets kind of jiggling around. I don't know if it's related.
Yeah, yeah, it's a great question. So we do see the droplets moving. We don't see them moving more significantly more when the pH is changing via diffusion. What we think might be happening here, and this is data from Thursday. Okay, so showing a different polymer and protein in the Coacervate. So we think here the droplets are initially pinned on the surface, and that perhaps then once the pH changes, material is leaving, potentially unpinning them from the surface, and then that's why they're moving. But yes, we want to look at all of that and see what's going on. But I don't even have the pH versus time plot for this yet. Yeah.
Thanks a lot for a beautiful talk. The experiments you showed evolve over time, and you also highlighted the interest of having non-equilibrium steady states. Have you thought about how to provide reservoirs to generate steady state type of behaviors?
Yeah, so we have thought about that. So mostly what we've thought about is having an actual chemostat, to where we can, flow in, set up an actual steady state system where we have a PID controller to control the pH and have that coupled to the diffusion of, or not the pumping of substrate in, such that we can ideally keep the substrate and pH externally constant. But we have talked about, so obviously we're familiar with Joop Bokoven's work. This is stunning. And, they have some really cool systems where their carbodiamide is, the DIC is insoluble, and they can basically have or immiscible, I guess. They can have a nice reservoir above. We don't yet have that system to be able to do that. But yeah, we're mostly thinking about setting up an actual chemostat to maintain the solution pH and substrate concentration.
Thanks.
Yeah, fortunately, we can buy all these enzymes and polymers, and so we can make a lot of Coacervate and actually build a small chemostat.
So I have a question regarding your measurements of the passive system that you mentioned at the very beginning, your elegant viscoelasticity. And so you mentioned that if I understood you right, the sticker lifetime is in the range of milliseconds, so 10 to the minus 2 seconds, and then the single chain dynamics would then be the chain dynamics between the stickers, so you're in the sticky rouse regime? Is
yes,
that what you're
yes,
observing?
That's right, 100% in the sticky rouse regime.
All
yes.
Right. Thanks very much.
Yes.
I still have 5 minutes.
Hey, very nice talk. I wonder in the biological system in the cell, what kind of molecules or structures are you trying to model with your system?
Yeah, great question. So there are two that come to mind. So one, our centrosomes have a very defined size. So we think about what sets the size scale of condensates in cells. So of course, a cell is not a simple, nice fluid that like we have in vitro. So there's an elastic matrix that could affect things. I think beautiful work from people in the audience. Obviously, you can have molecules that are interfacial stabilizers, and controlling the concentration of that could control droplet size. But it's also possible that reactivity could as has been shown theoretically, could control droplet size. So that is one biological model that we're looking at. But then also, there's evidence that the production of RNA and transcription then regulates condensate formation, which then has feedback on the rate of RNA production. And so also we think about that as like, I would say, a broad model, but has nothing to do with that I don't know. I don't know if the resolution of the imaging has been sufficient to look at that, but that's an interesting question for sure.
Right. A question about what you said about buffer. Did you mean to say that buffer suppresses pH change?
Yes.
No, I mean in your actual observation that when you added a buffer, then you are not able to observe the pH change that you expect. So therefore, all your observations are done in the absence of buffer.
Okay. Yes. Great question. So a lot of these experiments are done in water. But we have also done them in the presence of buffer. It just, so it can slow the rate of pH change. It can obviously then, it depends on the pKa of your buffer and how that relates to your starting pH and your final pH. So we have used that to try and better get an asymptotic pH change, where we start at some initial pH and then we use the buffer to control our final pH that we can achieve, and then we can better fit that to a sigmoidal pH change. Yes, the buffer does impact the pH change, and typically the amount of enzyme that we have present is not producing so many protons that like the concentration in that movie I showed, the concentration of glucose is on the order of like 100 micromolar. And so if you're producing 100 micromolar protons, in the presence of any buffer that most people would work with, a 25 millimolar buffer, like, forget it. That's not going to do anything to change the pH.
What an honor. Maybe on this question in comparison to biology, when I think about biological phase separation, I always think that everything is crowded with macromolecules. But here you have macromolecules that phase separate from a buffer, which is quite different in composition. Do you think that changes everything, in particular in terms of surface tensions?
Yeah, that's a good question. So we haven't tried doing anything like adding crowders. I will say for both of these enzymes, they're, in particular in relation to the GFP systems that we often work with, their partitioning is not so good. I mean, I guess it's still, very dilute relatively in the dilute phase, but at least there is still some enzyme out there. But yeah, that would be interesting to see how that might impact this behavior.
So let's thank Allie one more time.